the alumni distinguished professor of neurobiology at Washington University, and it gives me particularly an honor being an alumni of Washington University and to introduce him. He has got, as most of you can are pretty familiar with his work, he is currently the PI of the Human Connectome Project and has received numerous awards. Particularly, I think he is a fellow of the American Association of Advancements of Science, and I think a president of the SFN, so right, for neuroscience, am I right? Are you currently?
No, that was a long time ago.
Oh, that was a long time ago, okay. Anyway, but I had the distinct pleasure of working with David, and he was actually one of my examiners of my dissertation, so that was an interesting experience. We have him to blame. Yes, you have him to blame. And he's one of the few neuroanatomists who I think really understood topology and topology preserving transformations, and was one of the driving force behind developing of large deformation diffeomorphisms that I think the community is beginning to appreciate. But without further ado, I would like to hand over the stage to Dr. David Van Essen, and we're going to hear about the human cerebral cortex, arguably the most complex object in the universe, right?
Just about, yeah. Okay. Thank you, Sarang. Great to see you again. Great to be here. And a chance to talk to you about many of my obsessions about cerebral cortex structure, function, connectivity, development, evolution. And then I realized, talking to this group, I should highlight my strong interest in improved methods of visualization that you'll see some examples of. Can you hear me okay in back there? Great. Okay, so let's start at a very high level. How does the human brain actually work to make us unique as a species, as individuals, with incredibly different and diverse talents, but all too many of our population is afflicted by devastating brain disorders. So I think we can all agree that deciphering the brain in health and disease is one of the grand challenges for the century at least, and the millennium if our species is wise enough to survive for some more years. A little bit farther down in granularity, but still at a high level, I love numbers about the brain, so let's start with a reminder that each of us has a brain that has about 90 billion neurons. The dominant structure of the brain is, of course, the cerebral cortex, but it actually contains only about 20% of the neurons, and that's because it's exceedingly enriched in synapses, well over 100 trillion synapses, and it's interconnected. The cabling within the white matter contains about 100,000 miles of myelinated axons, plus even more unmyelinated ones. There's also the pint-sized cerebellum. I think it's a wonderful structure. I won't have time to talk much about it, but it is worth noting that while it's only 10% of the brain mass, its granule cells comprise about 80% of the total neuronal count, so it's a winner in that regard. Over on the right, we see loose pointers to the rest of the brain. Less than 10% of your brain mass, less than 1% of your total neuronal count, yet it's extremely important. It's hundreds of distinct little subcortical nuclei that are, some of them well understood, some of them still mysterious, but they are critically important for keeping us alive and well, handling our respiration, our appetite, and all sorts of other important things. So, how do we untangle the wiring of the brain? A tremendous challenge. Before we get into the nuts and bolts of that, I want to make a quick set of analogies with genomics, which has been, of course, a spectacular success of the late 20th and early 21st century. We have complete sequencing of the mouse and human genome. We know that in both of these species and in many other species, there's only a few billion base pairs and a strikingly small number of genes, the basic units of transcription, a little over 20,000 in us and maybe even a few more in the mouse. When we look at the brains of these two different species, we see the tremendous difference, literally more than 1,000-fold difference in the size of a human versus a mouse brain, and more than 1,000-fold in terms of the number of neurons there are. If we look at the basic units of the brain composition, the closest analogy we can make to genes, there are hundreds, as I mentioned before, hundreds of subcortical nuclei more in us, but perhaps not that many more. In terms of the cerebral cortex that we'll spend most of our time on, we know there are about 40 cortical areas in mice. And in the human, it's been a long-standing century-old debate. Is there only about 50 or there may be 200 or more? And we'll come back to that and actually provide a more solid approximation to a real answer. But before we get there, I want to spend some more time on background and context of cartography and connectomics in animal models, particularly non-human primates. I'll spend most of my time on giving you still a pretty high-level view of the Human Connectome Project with a focus on multimodal cortical parcellation we recently published. And then I'll wrap it up very briefly. So for me, it starts about 40 plus years ago when I started working on monkey visual cortex, and studies in those days were generally reported in terms of data presented on histological slices or drawings of those slices, and one slice versus another is very hard conceptually to stitch them together. And I realized that the cortex is a folded sheet and it ought to be possible to make a map. And I literally developed a pencil and tracing paper, crude but manageable for a while, method of making flat maps. We started with just a map of occipital cortex. Some years later, we made maps of the entire monkey cerebral cortex, but those were, again, for a long time using just the pencil and tracing paper. But on those maps, Dan Fellman and I, for example, could emphasize that it's possible to parcellate the visual cortex into 32 distinct areas based on differences in function, connectivity, architecture, and or topography. And then a comparable number of parcels in the non-visual portions of cortex. A small number of those areas or parcels were essentially poster children. They are well defined, like area V1 and MT, by any of these methods, or all of them together give the same answer, which is great when you can. But for most of these other areas, it was the case then, and it still is to a striking degree now, that the areas are fuzzily defined, debatable, the boundaries are subtle, there's noise and bias in the data. And so the current rendition is we can show you a computer-generated surface model of one parcellation from Paxinos et al. That has 164 architectonic areas. Markov et al. In a study I'll come back to reported 91 architectonic areas. We published an intermediate number, 130 or so, based on architectonics and retinotopy. But there's, again, not so much agreement in many of the regions, particularly the so-called cognitive areas of a monkey's brain. So cortical parcellation was and still is a hard but extremely important problem. It's a challenge. One of the reasons it's important is it gives us a framework for talking about the wiring of the cortex. And I started at this decades ago, starting to build hierarchical schemes of different cortical areas and how they're connected to one another. This was before modern pathway tracing methods had gotten fully rolling, and but a few years later, in the same study with Dan Fellman, we could generate a hierarchical model schematically showing the connectivity of 32 different visual areas interlinked by hundreds, several hundred wires, connections from one area to another. This is the most well-known representation, the so-called subway chart of the cerebral cortex. But we took the same data at the same time and put all 305 areas, filled in the dots or the pluses on a 32 by 32 connectivity matrix. And this was, in retrospect, the first parcellated connectome, though it certainly wasn't called that for many years to come. One of the things that was frustrating at the time is we knew full well that some of these pathways were very strong, like V1 to V2, some were very weak, but we had no decent quantitative data, and we started working on that. But the major advance to this date is from a pioneering study by Nicola Markov in Henry Kennedy's lab, who put tracer injections into 29 different areas in separate experiments. here's one example, injecting cortical area, parietal lobe, cortical area 7A. The tracer distribution from retrograde labeling revealed a very complex pattern that was analyzed quantitatively and then put onto this surface map. And this example shows that there are well over 50 different cortical areas nearby and far away projecting back to area 7A, and that wasn't an exception. On average in this study, there are 55 different cortical areas providing inputs to an injected cortical area. So the connectivity is far richer than Dan Feldman and I had appreciated. The other fundamentally important principle, one of the takeaways from this presentation, I hope, is that the strength of connections not only varies from strong to weak, it varies in a systematic, mathematically describable way as a log-normal relationship. A few strong pathways, most are intermediate, but they decline into very sparse pathways. They're all players in the game. What it all means is still to be worked out, but at this stage we can summarize our understanding of the monkey's cerebral cortical wiring diagram by this 29 area injected by 91 area pathways, and there are about, there were over 1,600 pathways identified in this one study. If you extrapolate to the remaining cortical areas and all of the subcortical areas I've alluded to, there's got to be at least 10,000 pathways defined in this way in the monkey, and our brains are surely even more complex, so it gives you a framework for thinking about what the Connectome Challenge actually puts up for us to work with. So with that as a deep background, let's now move on to the Human Connectome Project, or HCP as I'll be calling it. I'll give you an overview and a summary, and then we'll work through some specific issues. So before we get in fully immersed in the human connectome, let me put this into the broader context of connectomics. A connectome is a comprehensive map of neuronal connections, just like a genome is a comprehensive map of all the base pairs. But we have to put this in big-time quotes because there are three different worlds of connectomics that barely talk to one another at present. So there's the microconnectome world. I saw some nice examples of people working on this realm here today, where electron microscopy or other ultra-high resolution methods allow one to identify and link up all neurons, axons, dendrites, synapses, glial cells, and make an accurate three-dimensional reconstruction, but it's hard, and no one's come even close to getting a cubic millimeter of microconnectome data. That's a target, but a big challenge. Then there's the mesoconnectome world that in animal models, particularly the mouse, but you saw some from the monkey already, where tracers, retrograde and or anterograde, are used to inject one or multiple places in a single brain and to trace local and long distance connectivity fairly accurately, but at the sub-millimeter, but not totally micron level. But these are extremely informative, but they have to be done on many different animals and combined together. Then there's my current world, the macroconnectome level, which can get comprehensive data in individual living human brains, but only down to the resolution of the individual voxels, which are generally, as you'll see, a millimeter or two on a side. And so each voxel contains hundreds of thousands of neurons or axons and millions of connections and synapses. So with that in mind, the NIH, we were deeply appreciative, decided to fund the Human Connectome Project, which started in 2010, and our target was to get 1,200 subjects, and we actually achieved that objective, and getting healthy young adults, twins and their non-twin siblings, so that we could look at heritability issues. And we did this using improved scanners. The hardware was better, and much improved pulse sequences that I'll touch upon. And we got 4 hours of scan data per subject, which is a lot of multimodal data, and you'll see the examples of these. And the data quality, we think the field generally recognizes this as exceptionally high quality data. We did get 1,200 subjects, 1,100 with good neuroimaging data. The data acquisition was completed a year and a half ago for the 3 Tesla data, and a little bit later for the almost 200 subjects scanned in Minnesota at a high-power 7 Tesla scanner. We got hundreds of measures, subject measures as we call them, on behavioral and demographic data. I'll come back to that later. We got magnetoencephalography from our St. Louis University colleagues on almost 100 subjects using task and resting state MEG. We got the blood for genotyping. We've just completed that, and that's going to become publicly available later this spring. Then we analyzed the data up one side and down the other. We're using improved pre-processing pipelines. I'll come back to that. Using better visualization methods. Our primary platform that you'll see many examples of is from the Connectome Workbench, freely available software. And then we've carried out a number of advanced analyses, some of which I'll have time to highlight. A critical objective that we fully endorsed was to share the data and not just dump it out there in a giant digital warehouse, but to make it user-friendly available to share the data and the methods and tools that were used. So the workhorse primary database is Dan Marcus's Connectome DB based on the XNAT platform that got modified extensively to expand the modalities it could cover. We made multiple data releases, in one including our final big data release just 2 days ago after a long slog to get the data there and get it and keep it clean and well organized. That you guys, I think, understand the challenges of complex data analysis and release. But in the years since this flood of data came out to the field, more than 7,000 investigators have bought into it in terms of signing on to the data use terms. You guys are not intimidated by petabytes, but we're still kind of pleased that more than a dozen petabytes of data have been shared by downloading and or acquiring what we call Connectome in a box that we ship to different institutions. We've released a lot of extensively analyzed data. Hundreds of publications are emerging, and this is still the early days, I think, of people making good use of these data sets. It's not simple. As you'll see, there's a lot of complexities to it. We've held HCP courses for 2 years, and we have another one coming just before OHBM in Vancouver. And if you can't take that lovely trip, the course materials are available on the website for a free download. This is our advertisement for the coast, and we encourage anyone who's seriously interested in rolling up sleeves to avail yourself of this opportunity. Much of the course is organized around the common themes that were brought together in a publication that Matt Glasser, whose name you'll see more of, first authored, what we call the Human HCP's neuroimaging approach. And there are 7 core tenets, and I'm going to run through fairly quickly, but we think they're pretty important. One is get as much data as you can. More is much better for many aspects of neuroimaging. And get it at the highest possible resolution that your situation, your scanner, and your time constraints allow. But things like multiband acquisition for fMRI and diffusion imaging gives you more bang per buck or per minute of scan, and that's very useful. I'll come back to that. Once you've got the data, please minimize the distortions of the data and avoid blurring of data that gives you superficially, it's smoother, it's cleaner, it's got better signal to noise, but you've paid a price, and I think many of you guys know that. But it's hard to overemphasize that to a field that tends to still has some bad habits. Another thing I'll key on is to respect the geometry of different structures. And basically, as you'll see, the cerebral cortex is topologically a sheet of tissue, and that needs to be respected if you're going to get best mileage. But subcortical structures are blobs, and they get a different treatment. Aligning the data across subjects has been a huge challenge. You'll see some exciting progress we've made on that, but a precise alignment across individuals and across studies is critical for making quantitative, careful comparisons. Using your results to analyze them in the context of an accurate brain parcellation is another fundamental, and we now have a better parcellation to offer the field. Once you've done it, follow the HCP mantra of make the data freely available to the extent feasible, including the extensively processed data. I'll show you an example of that as we get towards the end. So this HCP style paradigm is applicable to other projects, some of which are already ongoing, the so-called lifespan development and aging and baby connectome projects. Another set, the connectomes related to human disease. There's 13 funded, hopefully more to come. A great big development childhood project, ABCD, and individual investigators and R01 subjects, like some I've already heard about here, can capitalize on these approaches. So what's the hullabaloo about these improved data acquisition and the different multimodal analysis? It starts with structural imaging. So this is a conventional T1 weighted image, except it's higher quality than is typically acquired. The resolution is 0.7 millimeters, and it lets us see not just that there's a cortical ribbon, but to turn free surfer loose and to use both the T1 and T2 weighted image data to make very accurate segmentations, capturing the shape of the gray matter and turning it into cortical surface models, where this is what we call the mid-thickness surface, which is midway between the white and peel surfaces. And we can not only generate that, but we can inflate it and show the folding pattern. The darker it is on this inflated model, the deeper it was in the original 3D volume. So what can one do with these structural images? There's already the ability to get interesting information related to function. And my introduction to this came when Matt Glasser arrived in my lab now almost a decade ago in 2008. He was a man with a mission. He had worked at Emory in a neuroimaging lab, and he had observed that in a conventional T1 weighted image, like this coronal slice here, the gray matter ribbon was indeed gray, but in some regions it was a little brighter than average, in other regions it was a little darker. He also noticed that in the same slice of the same subject, in the T2 weighted image, the region that had been brighter is now darker in T2. The region that had been darker is now brighter. So he did the math, divide and conquer by taking the T1 divided by T2 ratio. Nothing fancy there, but with doing that and doing some fairly basic corrections for bias fields and a little bit of fancy footwork, he was able to colorize this and to paint it accurately onto the individual subject cortical surface model and show that the patterns of where the ratio was high, we colored red, and that correlated well with histologically defined estimates from a century or so going forward with myelin content. So this is the well-known sensory motor strip that's heavily myelinated. This is the MT complex, known to be heavily myelinated, as is primary visual cortex. Primary auditory cortex, primary visual cortex. So this correlates well with cortical areas and functional organization. And it's not the only thing that correlates interestingly. We can go back to the surface reconstructions, and FreeSurfer automatically computes cortical thickness, which is hard to estimate on a slice-by-slice level because of obliquity of sectioning changing all over the place. But one can map that accurately onto the surface models. And here is a group average thickness map, which shows some regions like insular cortex are very thick, other, like primary visual cortex, are very thin. The early sensory areas tend to be thin, and they tend to be heavily myelinated. And interestingly, these early sensory areas also at a cellular level, by completely separate studies done by Guy Elston and colleagues, these early sensory areas have smaller cell bodies and smaller dendritic arbors. They're just packaged more tightly. Whereas the higher cognitive areas tend to have thicker cortex, be lightly myelinated, and be populated by bigger neurons with more robust, broader dendritic arbors. We don't think this is a coincidence. We think they're mechanistically linked. I'll touch upon that briefly in a few minutes, but I want to move on. We've covered cortical structure and thickness and myelin. Now let's move on to function and connectivity related measures and introduce this multi-slice or multi-band imaging that our colleagues at Minnesota, Camille Ugerbill and colleagues, as well as the Mass General group, pioneered this idea of, instead of tediously chunking through the cortex one slice at a time, excite up to 8 slices or more at a time, and use parallel imaging methods. We use the standard Siemens 32-channel head coil. The receive coil array can unalias these images and reconstruct the scene of the crime, as it were. And we use a multiband factor of 8 for the HCP's fMRI data, smaller but still beneficial 3-fold multiband factor for diffusion imaging. It gives a whole lot more data as well as demonstrably better data when you look at it carefully. And, but I want to come back to this point of lots more data. That's great, but you got to crunch through the numbers and you want to do it efficiently. And one of the efficiencies we realized early on was that for fMRI, one cares mainly just about the gray matter voxels, the cortical and subcortical. All the white matter stuff is largely irrelevant. So why load up your data sets with that inefficiently? And when you're dealing with the cortical structures, for reasons I've already, I hope, made clear, it's really important to use a surface-based model, a topological respecting vertex-described surface model. And so we use a single data format, we call it the SIFTI format, that can represent just the gray ordinates, the surface vertices for cortex, voxels for subcortical blob-like nuclei, and do it all very efficiently, and set yourself up for doing surface registration for cortical structures, volume-based registration, but nonlinearly for subcortical alignment. And if you're going to do any smoothing, there are some places where a little bit of smoothing is reasonable. Do it in gray ordinate space and respect the cortical topology and subcortical area regional boundaries. We'll come back to fMRI in a moment. I want to say a little bit, I don't have time for much, but a little bit about diffusion imaging. An exciting, a very important, but a challenging modality. As most of you probably know, diffusion imaging relies on faster water diffusion along the length of axons relative to crossing axonal membranes. And that information, by probing at many different angles, high angular resolution, Hardy in particular, lets you estimate fiber orientations, not only of the dominant fiber bundle orientation, but also the evidence for crossing fiber bundles, which are rampant in many regions of white matter. For the Human Connectome Project, we were able to use an improved scanner that gave stronger gradient strength, which is critical for getting the best diffusion imaging. As I mentioned, a multiband factor of 3 let us get voxel size 1.25 millimeters, much better than the typical 2 millimeter, and more fiber angles and better probabilistic tractography, where one strings together the estimates from individual voxels and estimates trajectories over long distances as best as one can. We've improved the visualization options in Connectome Workbench. So here is a seed for looking at tractography connectivity in the inferior frontal gyrus. And here is the fiber bundle probabilistic trajectory going through deep white matter within an anatomical framework that can be flexibly worked with. And then one can measure the tractography-based connectivity estimate locally and at long distance and see evidence for structural connectivity, as we call it. And while the HCP diffusion data and the tractography data, we believe, is much better than conventional scans, and we can get gray matter to gray matter dense connectivity, there are lots of false positive and false negatives for reasons that are at least somewhat well understood, but very challenging to work around. And we can talk about that later if people have questions or talk about it offline. But let me just jump over and spend more time on the modality which at this stage has proven more informative on a variety of questions for reasons you'll see. So this is resting state functional connectivity. A subject is in the scanner. with eyes open but no particular task assigned, but the gears are turning. What's for dinner? Where are the kids? And all those things that are freewheeling thinking, and they lead to very complex spatiotemporal patterns of the bold signal that we can pick up in the scanner. Functional connectivity, the basic concept is simple. Take every gray ordinate and look at its time course and correlate it with every other gray ordinate. Are they strongly correlated, weakly, or anti-correlated, or what? And that leads to a 90,000 by 90,000 dense connectivity matrix, about 30 gigabytes worth of data in this SIFTI-based format, and we can interrogate that one row or one slice at a time by making estimates of seed-based connectivity. So a seed location number 1 shows a correlation pattern, the functional connectivity, that is strong locally and in the opposite hemisphere and way off in the frontal lobes, not unlike what you saw from diffusion imaging, but they're similar but not identical. A different seed location gives a different pattern, and that's the nature of functional connectivity using the seed-based approach. We'll come back to that, but another important approach to looking at functional connectivity is what our Oxford colleagues Steve Smith and all have identified using independent components analysis, or ICA. So basically, here is one ICA node from a group average. It's a spatial map of regions that are strongly correlated with one another, but they don't have to be, they're often spatially nearby, but they can be off at a great distance. That's one ICA node. Here's another ICA node, one that's related to the so-called task negative or default mode network. I'll come back to that. But one can take a set of ICA components at whatever level of granularity you've chosen. Here we have a 25 by 25 network connectivity matrix, or NetMat, and we can do a full correlation analysis on that, which has some warts and wrinkles I don't have time to go into. Many prefer to use partial correlation as a way that cleans up the data and may give a better estimate of actual connectivity. But whatever decisions are made on that, we can now start to ask the questions of how does functional connectivity relate to things we care about, like behavioral phenotype. Well, recall that I said we had almost 500 behavioral measures. Many of those came from the NIH toolbox. Others came from a battery of neuropsychological tests. They cover a wide range of phenotypes: cognition, motor, sensory, personality, intelligence, environmental factors, plus demographic and psychiatric information. Some of this is restricted data, so you have to dive deeper to get it, but it's all available. We have 476 subject measures. If you look for any one of these measures, like fluid intelligence, and say, how well does that correlate with this pattern of functional connectivity? That's where we started, but we weren't sure what to expect. What we found is that the data are noisy, even after all this high quality and tons of data. It's hard to find really robust correlations between functional connectivity and individual subject measures. That's just the reality of working with a complex brain with the tools we currently have. But taking those tools and applying more sophisticated analysis methods brings good outcomes. So what Steve Smith and colleagues did was a canonical correlation analysis, or CCA. I suspect many in the room know more about this than I do, but I know enough to say that it basically identified a mode for the subject measures that was highly significant, another mode for the connectivity, and the subject measure mode shows an axis that goes from basically positive characteristics like strong vocabulary, high intelligence, good life satisfaction, good oral reading, and other things we consider good things to have. And at the other end of that axis, tobacco usage, THC usage, drug problems, sleep issues, all those things that we would say better avoid those if you can. So those two modes the behavioral phenotype mode and the functional connectivity mode show a correlation that's very strong, and one of the measures, the fluid intelligence, you can see in individual subjects that there is a positive correlation there. So there is information about individual subjects, but it's related to a complex pattern of connectivity. So if we take the connectivity aspects that I just elaborated on, and just take a quick but somewhat deeper dive, this is the 30 edges or connection, functional connectivity correlations between 30 different nodes, and some of them are strongly positively correlated, some of them are negative correlated, and if we look at just the positive ones and say, where does that overall mosaic of 30 edges that have this functional connectivity component, they're found in regions that we think of as higher cognitive, but they're not the same regions as, they don't include everything that people have previously tied into fluid intelligence specifically. So in a nutshell, we think there's a lot to mine using this kind of approach. We're going to reanalyze the latest release of data and hopefully get cleaner, better data, but this is an approach that is at least a good starting point. I'm going to move on, though, to continue our survey into a bullet that has three important and interesting and interrelated aspects: individual variability, cortical development, and intersubject alignment. So keep your seatbelt fastened. Let's look first at this issue of individual variability. So here are 4 individual subjects, right hemisphere lateral views, and if you look closely, it's a little hard to tell, but they don't look the same. They're folded rather differently, and that's been known for a long time, but seeing it with your own eyes is informative, and it's more informative if you take these same surfaces and inflate them as we've done before, show these maps of sulcal depth, and notice that there are some regions, while the regions like the central sulcus are quite evident in every subject, regions like this prefrontal, inferior frontal sulcus, it's folded very differently from one individual to the next. And that is true even when you look at identical twins. So as it happens, there are 2 pairs of identical twins here. Can you tell on the fly which are genetically driven to have more similar folding patterns? It's really hard, but these are 2 twins. These are 2 MZ twins. They are correlated in a way that shows some heritability. It's not 0, but it's not as much as you might have guessed. So that's one very important aspect of individual variability. Here's another one. The cortical areas that I've talked about in monkeys and will return to in detail for humans, but some of them have been well defined for a long time, like primary visual cortex. And if you look at individual maps of V1, like this red patch in different subjects, you see that it varies in size and it varies in its location relative to these folds. So here's another fundamental aspect of cortical variability. So let me step back from that for a moment and say, well, wait a minute. The cortex, sure, it's convoluted. Why is it so convoluted? How did it get that way? And while we're at it, why is the cortex a thin sheet? What makes it that way? And why is our brain, while it's convoluted, have such a wildly different convolution pattern compared to a camel or an elephant? This is just an amazing aspect of mammalian brain structure. There's not a single formula, but maybe there's similar mechanisms across this diversity of outcomes for different species. And I propose that there is, and here's my thoughts on it. First of all, why is the cortex a sheet? Here it is in a coronal view of a very early stage of gestation. The ventricles are like water balloons. They're very large because the choroid plexus is pumping out fluid and keeping it inflated. Along with that pressure to inflate the cortical mantle is the fact that the developing cortex is dominated by the apical dendrites of pyramidal cells. And here you see drawings of individual pyramidal cells. That apical dendrite comes first, and it dominates even well into maturation. So suppose that those apical dendrites, or that all dendrites dominated by the apical dendrites, generated just a little bit of mechanical tension, that they were trying to keep themselves short even as they got pulled apart. If that mechanical tension were present, then this anisotropy of the architecture would lead to the cortex trying to stay thin, and as it grows, it would just expand in surface area, enhanced by the water balloon mechanism. So if that's the case, what about these couch potatoes over here, these subcortical nuclei? Why are they just big blobs? And my hypothesis is it's their cellular architecture. If you look at them, this is schematic, but it's the reality that subcortical nuclei tend to have isotropic or quasi-isotropic dendritic arbors. And so if they're generating tension, they're trying to stay compact, but not sheet-like. So here's a simple hypothesis supported by the fact that neurites in tissue culture do indeed generate hefty chunks of mechanical tension. So it's a plausible hypothesis for why the cortex is folded, and now, or why the cortex is thin. Now let's turn to the question of why is the cortex folded. And the first observation that gets us into the right framework is to note that maturation in the third trimester goes from a very smooth or lissencephalic cortex at the beginning, and by the end of the third trimester, the cortex is almost as convoluted as in the adult, even though it's only 1/3 of its eventual size. So there's a lot of growth, but the folding is largely in place. And so my hypothesis for why the cortex folds comes back to mechanical tension, but now we focus on the axons instead of the dendrites. So the dendrites were, by hypothesis, what makes a cortex thin. The axons going through white matter, here in reality, here in the schematic, would cause cortex to come closer together if it's linked by regions that are strongly connected by coherent bundles of axons. So this could be V1 and V2, for example, coming closer together by virtue of coordinated pulling, tugging by those dendrites. So this is a hypothesis that accounts for a gyrus as the outcome of a competition with strongly connected regions being linked. And a sulcus is essentially a losing battle that perhaps because of weakly interconnected regions. And variability of the folding from one person to the next could reflect variability in the size of different areas and or their connectivity. So there's a rational framework for thinking of that as well. So it has been for now 2 decades an attractive hypothesis. It's absolutely not proven. It's almost certainly not the only mechanism in town. I don't have time to explain the others, but differential proliferation deep in the ventricular zone and cortical buckling are probably also players. So as in many aspects of development, there are multiple mechanisms or players, but here's one to be thinking about as part of the equation. With that in mind, let's return to adult subjects and grapple with this issue of individual variability in the folding pattern and in the relationship between cortical areas and cortical folds. So the standard-based method of surface-based registration that my lab started with and Bruce Fischel's free surfer method has put forward is to use the folding pattern itself to constrain the registration across subjects. And when that is done, one gets pretty good alignment of primary visual cortex, tolerable but not nearly as good alignment of area MT and various other areas. And basically, we have long felt that folding alone is not sufficient to drive accurate inter-subject registration. We needed a new method for doing that. And fortunately, our colleagues in England, Emma Robinson and Mark Jenkinson and colleagues, developed the multimodal surface matching method that's based on discrete optimization. It's flexible in what features it uses. It's highly tunable. It's low sensitivity to local minima. And as I'll show you, it works very well on HCP data sets. But just not to get bogged down in details, but basically this multimodal surface matching, like other methods, uses registration on spheres and uses rotational control points and has recently been improved so that it uses point triplets, not just point pairs, to constrain the cost function and do good things. So we can talk about it afterwards if people care. But for now, I just want to make the empirical point that it works. So here to document that, here is an alignment of a task fMRI activation pattern, listening to stories versus baseline listening to nothing. And if one uses folding alone to align many subjects together, one gets a pattern of alignment that's not bad, but it gets much better, particularly in this language-rich region here, using functional connectivity to improve the alignment. Here's one more example, a prefrontal region that is better aligned, sharper, stronger signal to noise, using aerial features. The requirement is that one needs lots of functional MRI data. We don't know how much, but we suspect a half an hour's worth of data will be the opening requirement, or at least approaching that, for accurate inter-subject alignment. So that's one of the reasons we emphasize got to have lots of data. With that in our armamentarium, let's now turn to this human cortical parcellation challenge. A little bit of quick history. A century ago, Brodmann gave a classic map that had 50 cytoarchitectonic areas. Around the same time, the classic myeloarchitectonic experts, the votes, said, no, we think there's actually about 200 myeloarchitectonic areas in humans. And that dynamic tension persisted for more than a century. We got into play with an estimate of at least 52 surface mapped areas and extrapolated to say there could be as many as 150 or more based on evidence available a few years ago. Many studies have started using resting state functional connectivity, analyzing resting state networks, and coming up with networks or parcellations that in this case have 17 networks. Steve Peterson's lab reported evidence for 300 plus parcels. Another group has reported well over 200 parcels. But these unimodal parcellations don't agree with one another. They're not strongly symmetric across the two hemispheres. And they are progress, but they're not an endpoint, we would say. We felt, and by that I mean Matt Glasser leading the charge in my lab, that we needed better parcellations using multiple modalities, going back to the principles that Dan Feldman and I had articulated, and to use objective criteria based on spatial gradients on surfaces for identifying borders. And we wanted to be able to parcellate individuals as well as group averages, and we'll show you the product of that as we go through it quickly. The first point is that Matt took data from almost 450 subjects and divvied it up. He identified a parcellation group where he did the initial group average parcellation of 210 subjects. And this is the group average myelin map after feature-based MSM registration. Here is the myelin map from another 210, what we call the validation group, totally separate subjects. And look at the similarities of the myelin maps and all the details, like this little notch in the myelin map turns out to be functionally relevant. It's the boundary between upper limb and face representations, and it's visible in both data sets. And nearly all of the fine details are replicable. Similarly for task fMRI, this is a language-activated right hemisphere task activation in the parcellation data set. It replicates, as do many other features, in the validation data set. So this gave us confidence that, yes, these fine details are worth attending to. And when we attended to them, we used this gradient-based approach that I'll illustrate here for our poster children. A little area that we call 55B for reasons I'll momentarily explain, sandwiched in as a lightly myelinated region in between moderately myelinated frontal eye fields and premotor eye fields on either side. When we take the gradient of the myelin map, we see a gradient ridge that's not as strong as between premotor and non-motor domains, but it's highly statistically significant. And when we compare it with other modalities, we start with task fMRI. So this is this story versus baseline activation pattern I showed. the spatial gradient of it is very robust, and it correlates with the myelin gradient. When we look at functional connectivity, we can take an example seed in a language-related area, see a correlation with this same lightly myelinated patch. If we take an eye movement-related location in parietal cortex, we see it correlates with the eye movement fields and avoids the 55B. And the gradient of all possible seed combinations shows a gradient ridge that correlates with these other two modalities. So we have consistency across multiple modalities, and instead of inventing a completely new name, we call this area 55B because half a century ago, Adolf Hopf had identified on his version of a flat map a lightly myelinated region just in front of motor cortex. So this was our strategy that is very painstaking for each individual area, and Matt applied this to all of the left and right hemispheres, yielding 180 areas, some of which we think will be further subdivided, so we call them complexes. And this is the outlines of these areas. We can colorize this to give you a sense of the functional specialization. So this is just one aspect of what we know about function, but the regions that are strongly correlated with the sensory motor strip in their functional, resting state functional connectivity, they're greenish. The visual ones are bluish. The reddish ones are auditory. And the task positive and task negative cognitive regions are shaded light and darker, respectively. So we get a functional signature that is present in the left hemisphere and the right hemisphere. And even the language-related areas that we might have expected to be dramatically asymmetric, given lesion studies over more than a century, the 55B is similar in its functional signature, as is the perisylvian language area. So we see differences, but they're not nearly as dramatic as we might have expected. And we do also know, importantly, that there's a lot of functional heterogeneity, particularly in the somatomotor strip. So here's an illustration of essentially a sub-area related to the hand, the face, and the foot as exemplar subparcels within the somatomotor strip. We took 83 of these areas and said they are similar enough to what's in the literature that we can give them the same name. But for 97 of them, we gave them new names, sometimes splitting existing parcels, but this is fundamentally a new map of the cerebral cortex, and the gory details are published in a supplemental table if you want, or extensive results. That's for the group average. Now, can I go up against the stops? Okay, sorry to be running a little bit on, but there's a long story here. So individual variability. Typically and schematically, area 55B is oriented away from the upper limb representation. In some subjects, as you'll see, 55B has a different orientation relative to that subject's upper limb representation. And that poses interesting challenges. So let me illustrate with quick examples. This is the group average language network and focusing on this outline of 55B, which is lightly myelinated and is well away from the upper limb representation. Here is one individual subject. The data are noisier, of course, but it's a pretty robust patch there. It's lightly myelinated and has the same pattern. Here is a subject which, after the functional aerial-based registration, this patch did not get registered to the group average accurately because it has a different relationship. It's topologically differently aligned. Here's another subject, and so what we assert is in some subject, the organization of the cerebral cortex is different in a way that topology-preserving alignment cannot get the job done, and about 10% of our subjects had this characteristic in this region of the left hemisphere. Well, what's a feller to do? Matt was able to take a classifier algorithm that Carl Hacker had developed in Maurizio Corbetta's lab, a perceptron-based algorithm, and to apply it carefully and methodically so that for each individual and each area, the features associated with each vertex are correlated with the multidimensional, 100+ dimensional feature vector for the group average to say, is this vertex likely to be the premotor eye fields, and give it a probability map for that? What about area 55B? Another run of the classifier says area 55B is probably here. Another run says FEF is about here. A winner-take-all says, here's the final decision, folks, for this individual. The areas agree with the group average. But when we take one of these subjects with an atypical topology, we get a different outcome. Probabilistic area 55B is sitting way up here outside the normal pattern. It's a shifted map. And when we do that across individuals, Matt was able to get this classifier to work for nearly all areas in nearly all subjects, 90+%. And here's an individual subject's parcellation boundaries relative to the group average. And you can see one subject versus two subjects. Those are the differences. We have the ability to identify nearly all these areas and show that the areas vary in size, shape, and location, but the areal classifier works generally well. We can make a probabilistic map from this classifier of all areas in the 210P group and from the 210 validation group. The independent data sets, the outcomes look very similar, and when you make a winner-take-all maximum probability map, the colors align for the blue for parcellation, red for validation, purple when they agree, and there's 0.97% validation alignment with the group average. So, very quickly, but still pretty importantly, data sharing, I mentioned it early on. The Connectome DB data base provides easy access to the raw, unprocessed, and minimally preprocessed data. That's what Matt used to get his data to do his parcellation. Now that we have this parcellation and the very rich, highly analyzed data, they don't fit well into the Connectome DB database. It's not built for that kind of specialization. Rather, we built a separate database called BALSA, and we parked these data in the BALSA database, and we made it so easily accessible that you can go to the online publication, go to any figure you're interested in, link to the BALSA database, see a preview of what's available, and download that data set in a very short order to make it on your laptop or local computer. And there you can look in Connectome Workbench at representations, for example, that show you the annotation of all the areas on both hemispheres and see what those labels actually correspond to. And you can start doing what we call parcellated analyses. Instead of taking a fuzzy but highly detailed myelin map, you can parcellate it and take the average myelin content of each parcel, or you can do that for every modality and every feature and start doing high signal-to-noise, multiple data analyses of many sorts. Lastly, in terms of data, going back to different species, here's our brains, 3 times larger than a chimpanzee, 3 times larger than a monkey, another 100 times larger than a mouse. The convolutions vary, but we can start doing evolutionary comparisons by taking information such as myelin maps that you've already seen for humans. We have them now in chimpanzees and the macaque, and we can see area MT in all 3 species. We can see the prefrontal higher cognitive regions are present in all 3, but they're greatly expanded in us compared to chimpanzees, compared to macaques. So it sets the stage for next generation surface comparisons. So, at long last, to wrap it up, we're in the still early stages of a grand challenge of understanding human brain circuitry in health and disease. The HCP is already helping to elucidate brain connectivity and how it varies. We're seeing major discoveries and insights, but we're far from and may never get to a complete human connectome. We need to manage our expectations, be optimistic, but keep a very critical eye on all modalities of data, but recognize that the improvements in data acquisition, analysis, and sharing that I've described will give us a better understanding of diseases, development, and aging, and their diagnosis and treatment. So I can't say enough in the seconds that remain about an incredibly dedicated and talented group of well over 100 investigators and staff. I'll just mention a few absolutely key people. Camille Lugermill was co-PI for the Human Connectome Project. Steve Smith led a lot of data analysis. Deanna Barch led task fMRI behavior and a lot. And Matt Glassler, as I've already mentioned repeatedly, did a huge amount in terms of many different aspects of this project. So I'm happy to answer questions, and thanks for your attention.
Thank you very much, David. But I think Jeff has a question. Yeah.
Yes.
So we are really excited to use the Glassler parcellation for our own data. And what tools can we use to do that machine learning classification? So are there any resources that are available that we can apply that to other data sets that are not Human Connectome Project data but of similar quality?
The short answer is, how can I get the aerial classifier or that strategy in play for our own research projects? Be patient. It's coming. The aerial classifier algorithm is part of our suite of tools, most of which we have released. Some are not yet released. The classifier sits in code written by Matt on his laptop and local machinery. We're anxiously awaiting the conversion of that to HCP pipelines that can run on the HCP data and the other data sets. It's not a huge amount of work, but it's been stacked up in a very long queue of things that led up to this most recent release. So we'll release it as soon as we can, but realistically I would say summer or fall before it's likely to make it out for road testing by investigators, but it's just to do what we've learned is to do these things well and carefully with the personnel we have at hand. It just takes a lot of time, and we don't want to rush and be sloppy in the data we release or the tools we release. And so that's where we are.
Okay, Osama and then Chris.
A question regarding the connectome that you had in the Nature paper. Why did you not use the diffusion data to help the other modalities other than the false positive and false negative that
What's our excuse for not using that beautiful diffusion imaging to get it? Excellent question, and our answer is twofold. One is data processing. So we were retuning the diffusion imaging, going back to square one. We got a better eddy current correction algorithm that we finally pushed through. We got a better fiber orientation bedpost X algorithm operational. We have not yet run the tractography, probabilistic tractography, our favorite version, on the Human Connectome Project data. So part of it was we didn't have the data right at hand at the time Matt needed it. Another part is even if we'd had it at the time, we probably would not have used it because of these issues of false positives and false negatives. Some of these tractography limitations are related to correlations between streamlines and gyral foldings. Tractography streamlines tend to end on the crowns of gyri, and they tend to avoid the fundi of sulci. We know why getting a fix is a really hard challenge for the future, and if you don't have that fix and use this gradient-based approach that I mentioned, you're going to get gradient ridges that are related more to folding patterns than differences in structural connectivity. There may be ways around this in the future, but it was more than Matt felt comfortable taking on because it would have added a lot of noise. We are very interested in seeing how well this parcellation, what it tells us about tractography-based connectivity, if we use that for a parcellated analysis of tractography. That's very feasible, but that's our rationale for not trying to jump in and add that modality, as interesting as it seems in theory.
Chris?
Thanks very much. That was fabulous. I'm sure you
you have a very long list of to-do items. I wonder if you could tell us maybe a few of your highest priority. Is it multimodality registration? Is it more accurate DTI algorithms? Is it better segmentation? Is it, besides just saying yes?
Yes.
What are some high priority things you would love to see?
Well, certainly one of them that I touched upon before is getting this aerial classifier out, run on all of our data sets, because we only have 449 at the moment, and evaluated in the context of the existing young adult HCP. That's my #1 priority, but patience is required, as I've mentioned. Getting these to work on new data sets, sharing it with the community, and also running that on our lifespan data sets now that the cranks are turning on those, and seeing how they help us learn about maturation, aging, and various diseases. They should be more sensitive. Are they sensitive enough to be substantially more informative about disease connectivity? That's an open question of critical importance. I'm also personally, things I am particularly interested in but don't have the bandwidth to do it and jump into immediately. One of them is looking at the heritability of connectivity in individual subjects. Another is using the connectivity data that we have to explore explore whether that has can be not to prove, but to make a stronger evaluation of the folding-based, attention-based folding hypothesis. If strongly connected regions have gyri in between them, we ought to be able to see that statistically now that we have a data set that's rich enough to do that. So that wasn't well explained, but that would be very interesting. And then this whole issue of interspecies comparisons. I have a student working, Chad Donahue, is working on this monkey to chimp to human registration, because animal models are enormously important. They will remain this way over and above what we can learn from human neuroimaging, but we need accurate maps between monkeys and humans, and the tools for doing that are much better now than they were a few years ago. So that's my short laundry list.
Any other questions? Oh.
Have you studied the disease connectome initiative?
Sorry, what?
The disease connectome.
Have I looked at it? So these 13 projects, the NIH started the ball rolling by funding 13 projects that are looking at aging and schizophrenia and depression and various other R01 style projects. And those, they are funded to use HCP style data acquisition and analysis. They are in the process, they're still all in relatively early stages, so none of the data has reached our WashU Connectome Coordination Facility that will be housing all of these data sets. So it's still too early, but my fingers are crossed that it's going to be significantly informative. I don't think we're going to get a magic bullet and say, oh, you know, this suddenly explained the roots of schizophrenia. But there's a lot of theories out there, and we're data impoverished for a critical evaluation of one idea versus another.
All right, let's thank David again for a wonderful talk.